Okay, girls, we're gonna introduce the nervous system today. Okay, so we've looked at a few systems. We've looked at endocrine. We've looked at thermoregulation and osmoregulation. So this is our next system. Okay, the nervous system. All right. So true or false? Grade 11 biology. Okay, I don't know if you remember this. Do all animals have a nervous system? The answer is almost all. Can you think of an animal that doesn't have a nervous system? So it's an S. There's a famous cartoon character that is this type of animal. Yep, sponges. Yes, so SpongeBob SquarePants would not be able to talk because sponges don't have a nervous system. But here's an example of two organisms. Here's a hydra on the left. This is a flatworm on the right. They have a nervous system. So virtually all animals have nervous system except for sponges. Why do we need a nervous system? What does it do? Well, it's doing one thing right now. Actually, think. Yeah, so it allows us to think. What else? It allows you to what? Move, respond to the environment, right? It also allows you to regulate certain things in your body, like your heart rate, for instance. And you saw it, the nervous system played an important role with endocrine and thermal regulation. So it has many, many roles. And we'll talk about how the system works as a whole. So, like, for example, we'll talk about like something simple like this. Like, this is basically a little flowchart that shows you, three things, basically. One is the detection of a stimuli. So that's called reception. So a stimuli has to be detected. The stimuli has to be processed or integrated, and then something has to happen. And this basically is, you'll see, because what we're going to do in this chapter is I'm going to show you how a neuron works. Now, a neuron is like a nerve cell. It's the functional unit of the nervous system. If you remember, we looked at the kidney, we focused on the nephron, right? So we can understand how a neuron works, and then the nervous system is a collection of neurons working together. So this here, what you're seeing here, is a collection of a whole bunch of neurons working together, communicating with each other to detect a stimuli, process a stimuli, and then respond to a stimuli. Like, for example, like you see a lion coming towards you, what types of neurons would be detecting that? Those would be neurons in your what? Before your brain, neurons in you've seen the lion in your eyes, okay? We'll talk about how that works. And then those neurons have to fire a message, and those neurons have to that message has to get to the neurons in your brain, right? And your brain has to process the information, say, oh my God, or maybe something that I can't repeat right now because I'm being recorded. And then your brain has to fire a message to other neurons that control like muscles in your legs so you can run away. We'll talk about how that system works. So we're going to come back to the actual system itself. We're going to come back to the nervous system after we've talked about the neurons themselves and how they work. So we'll start at the basic unit, and we'll come back to this, okay? So I'm going to come back to all of this. So what I'm going to do is I'm going to focus, now there's different types of cells in the nervous system, but the one I'm going to focus on are the neurons. There are other cells like glial cells that assist neurons, okay, and we'll talk a little bit about them, but the one we're going to focus on is the neuron, okay? And there's three types. There are the sensory neurons that detect stimuli. So give me an example of a sensory neuron, other than the one I just mentioned, the one that detects like something visual, which, by the way, would be the only type of neuron that would be. What type of reception? That would be like photoreception. the ability to detect light. So photoreception is the ability to detect light. What do you think thermal reception is? Yeah, temperature. Yeah. So you guys learned the basic senses in elementary school, right? The 5 basic senses. So, those senses are detected by specific neurons. So what would be an example of one? A neuron that allows you to detect what? Is that? Taste. Yeah, very good. Yeah, very good. And where would that reside? Where would that be? Yeah, in your mouth, in your tongue, right? Okay. Then there are motor neurons. These are neurons that allow you to move, because the motor neurons, they control movement by stimulating muscles. Okay. And then there are the interneurons, and the interneurons are the ones that connect the sensory and motor neurons. And I'll show you that in a second. Okay. And by the way, you see the word afferent and efferent. So an afferent, that means that the information goes towards the interneuron, and then efferent means the information comes away from the interneuron. You'll see that, actually that was in this slide, I believe. Yep. So you can see the sensory neuron, for example, something that you would taste. So there's a neuron that would allow you to detect taste. You can see it. Afferent means towards the central nervous system. Your brain would register that and say, "Oh, that tastes really good." And then it would send information, efferent neurons, to your muscles. Say, "Go get some more food." Right? Okay, so it's an example of like your neurons working together. Here are the 3 types. Here's a visual of them. So this would be a motor neuron, okay? It controls muscles. Here's a sensory neuron that allows you to detect things like light or temperature or pain, okay? And here is a brain cell. This would be an example of an interneuron, a neuron that processes information, right? Here is probably the simplest of systems working together. This is something called a reflex arc that involves basically just the 3 types of neurons. So you have a sensory neuron here. You have an interneuron right over here that processes the information. And then you have a motor neuron that responds. So what do you think is going to happen in this case? What would the sensory neuron detect? Heat, right? Fire information to the interneuron. The interneuron would say to the motor neuron, what? Tell that muscle to contract and pull the arm away. Okay. A reflex arc, by the way, is a very simple system. It's an example of one that you don't have to think about. Like, for example, I'm sure this happened to you numerous times where you touch something really hot or you touch something very sharp, and what do? You automatically pull your hand away. Do you think about that? Do you think, maybe I should, as my hand is cooking and I start to smell it, do you think I should pull my hand away at that point? No. So reflex arc is something that is really quick because it doesn't involve a lot of neurons. The more neurons it involves, the slower it is. Like, think about how simple this is compared to if I ask you a question like solve 10x minus 7 equals 100, for example, right? Then that takes a little bit more work than, so that means it involves more neurons than something like this. Okay, here's just another, this is the same picture basically, okay? Here's a reflex arc. You'll notice that 2 of the 3 neurons, so here's a neuron here, here's another neuron over here, and here's a 3rd neuron over here, right? There's your sensory neuron, your interneuron, and motor neuron. 2 of the 3 neurons are coded, so they have this code, which I'll talk about in a second in the next slide, okay? not all neurons have this coat. You see this coat here? This coat, which is called the myelin sheet, to me it kind of reminds me of the plastic on a cable. So, like, for example, if you look at this cable here, you'll notice that it's coated in plastic. Now, if you look inside a cable, what kind of material would be inside a cable? What is it? Yeah, it's some conducting material, maybe like copper, for instance, right? Why a conducting material? Because what's running through the cable? Electricity, right? Very good. I need you to think of the neuron the same way, okay? Because you'll see that in a second, that it's almost like the movement of electricity down the neuron. The coat protects that flow of information, but not all neurons have it. So you can see that there is no coat over here, okay? So things like sensory neurons and motor neurons would have these coats. Now let me ask you a question then. What if you had a disease, and we'll talk about diseases, that maybe deteriorates this coat? What would happen to that person? What do you think? So there's a disease called XALD. There's a movie called Lorenzo's Oil, which I show my students sometimes. It's a very sad movie where the condition leads to the destruction of the myelin sheath, which is this coat. What do you think happens to that patient? Yeah, so there's going to be, the information will be disrupted. And you'll notice the motor neuron, we talked about, controls what? So what would you see in this patient? Maybe the movement will be affected, right? Maybe you notice that the person's walking a little bit funny. Maybe they trip. Maybe they don't look like they're walking normally, right? I said walking twice now. But you'll notice something in the way they move. Because as this deteriorates, what happens then is the signal to the muscles deteriorates too. And we'll talk more about diseases later on. Okay, so here's like the basic anatomy of a neuron. Okay, so it's basically divided into three parts. So over here we have, so if you look at this section, this is the front end of the neuron. Okay, I need you to think of this part like the ears or eyes of the neuron. This is the part that receives information. The ends over here, these ends, are the parts that send out information. So this receives, so inputs, and this is the output for a neuron. This then is the cell body. Okay, the cell body is, well, this is the cell, right? So what would you find in here? You would find the nucleus, you would find the Golgi body, the mitochondria, the ribosomes, okay? This then, the way a neuron works is it receives information here, it processes the information, then it spits it out. And we'll talk about how this works. And there's that coat, okay? Now you'll notice that there's like gaps in the coat, and we'll talk about, you can see some gaps here. We'll talk about the significance of those gaps in a little bit. By the way, the coat is actually a cell. It's actually a supporting cell called a Schwann cell or a glial cell. And these cells are the stuff that acts like the plastic around the wire. Okay? So it's actually, this is a cell that's coated by another type of cell. So here we talked about the dendrites, the cell body, and they talked about the myelin sheath. Now, do you guys know what a nerve is? Because often we confuse nerve with neuron. A nerve is just a collection of neurons. Okay? So here is a cross-section of a neuron. So you see all these little, they look like little dots. Okay? These little dots are the tails of the neurons, which are called axons. So if you were to go back to your picture of a neuron, the neuron you see has a tail. That's where the information comes out of. The tail is called an axon. So this tail is called an axon. So what you're seeing here are all of the axons from the neurons. So a nerve is a collection of many neurons. So when you sever a nerve, you're not destroying one neuron, you're destroying hundreds or thousands of neurons. The bigger the neuron, the bigger the nerve, the more neurons it has. So, for example, your spinal cord, right? The spinal cord has a lot of these nerves running down the spinal cord. What would happen if that got damaged? Someone with spinal cord injury, can they move? Not very much, right? Depending obviously on the severity of the spinal cord injury. Okay, now, what we're going to talk about, I'm going to focus really on three things when we talk about the nervous system, okay? Because what I want you to understand is I want you to understand how a neuron works. Okay, that's one. How a neuron works. The other thing I'm going to talk to you about is how neurons communicate with each other. And then the third thing is I'm going to talk to you about how the neurons get triggered. So there's three things, right? One is basically how does a neuron fire information or release information? And that's called an action potential. All right? And the second thing is we'll talk about, and we'll talk about today, okay? The second thing we'll talk about is how neurons communicate with each other. And then the third thing we'll talk about is how a neuron gets triggered in the first place. Okay? So those are the three basic ideas. The three most important ideas in this chapter. So first off, you have to understand what the term polarization means. Something's polarized. What does it mean? It's polarized. That means one side is different from the other side, okay? So for a neuron to be triggered, okay, and fire off information, it needs to be polarized. In other words, the outside and the inside have to be different from each other. And that's what you're seeing in this picture. So what's the difference between the outside and the inside in this picture? What's the difference? A lot of sodium on the outside and a lot of potassium on the inside. Guess what neurons have? They have a special pump that you saw in the kidneys that does this, that establishes this difference. What do you think the name of that pump is? It's the pump that aldosterone works on. It's called the sodium potassium pump. Okay? Now, when a neuron fires, it's generating what's called an action potential. Okay? So an action potential, or we can call it a pulse, is basically the movement of those two ions, sodium and potassium. Okay? And what we're going to do today is, so we're going to focus on three things. Today we're going to talk about how does that work when a neuron's firing a pulse. What does that look like? So here's an example of a neuron firing a pulse. This could be, for example, the detection of odor. Okay? You smell something, a neuron fires information to your brain. We're going to look at how that works. Okay? This is the trigger mechanism of how that works, but I'm going to come back to trigger mechanisms later on. So I'm going to tell you something right now, okay? That's very important. So listen up. All neurons fire the same way. So whether I give you an example of the neuron that detects light or smell or temperature or pain or thinking, they all fire the same way. But what's different is how they get triggered. Okay? So the trigger mechanism is probably different, but the way the neuron works is always the same. So in other words, the difference between a neuron that detects odor and a neuron that detects pain is the way they're triggered. Okay? So A neuron that detects odor has a receptor for an odor molecule. There is no receptor for pain. There's a different mechanism. But the way they work is the same. So let's look at how they work, okay? This is the basic idea. Has to do with the flow of those two ions, sodium and potassium. So this is the cell membrane of the neuron, okay? You'll notice that there are voltages here in this diagram, okay? Because there are two types of gates. There are what are called voltage-activated gates, and there are the gates that are not controlled by the voltage. They're passive. The voltage-activated gates will open and close depending on the voltage. So here's the sodium gate. You'll notice that at negative 70 millivolts, which is considered like the resting state of the neuron, the gate is closed. But if we could increase the voltage to negative 55 millivolts, the gate swings open. Now this is an important piece of information, okay? This is a picture of an action potential. Have you ever seen this before? Where have you seen this image before? Maybe you see like a movie, or a show with a doctor show like ER or something like that. You see the patient being hooked up. This is like what the heart rate monitor would show you, right? EKG. Because the way a muscle works is very similar to the way a neuron works. It's the flow of ions, so it's going to give you the same pattern. This shows you the change in voltage of a neuron when it's firing, okay? When it's doing something. You'll notice that the voltage starts off at so here's your minus 70 millivolts, okay? Here's your minus 70. And you'll notice when a neuron is firing, you can see the voltage goes up, and then the voltage comes down. So when a neuron's firing, the voltage goes up and down, up and down, up and down, up and down, okay? So we'll talk about the trigger mechanisms. We'll talk about what actually gets the voltage to fire. Gets the neuron to fire. But today we're going to talk about how is it firing? What's it doing? Okay? So let's start with this picture. Okay? Most of the sodium is on the outside, and most of the potassium is on the inside. When a neuron fires, the voltage goes from minus 70 to minus 55. And at minus 55, the sodium gates will open. So what happens then, when a neuron is triggered, the sodium gates will open. Now what happens? Well, if there's a lot of sodium on the outside, it's going to want to diffuse into the cell, right? And you can see that here. But now we have a problem, because potassium, not only does potassium want to get out, because there's more of it inside than outside, but what happens when you put a lot of positive things together? Do they like that? No, they get a little bit angry, right? They want to leave. So what happens then is the potassium gates will swing open because of the voltage change. So at this point, right over here, you'll notice that the voltage changes. It goes up, right? And then it comes down. The reason why it comes down is because the voltage increase causes the potassium gates to open. Now what happens? Potassium exits. When potassium exits, then you have this scenario. You have now a lot of sodium on the inside and a lot of potassium on the outside. So you can see why the voltage goes up and down, right? Why does it go up initially? What's happening on this side? What's rushing in? Sodium. What's sodium's charge? Positive. So if you have something that's negative and you start adding positive charges, which way does the voltage go? Towards the positive, right? Now, what happens on this side? Potassium exits. And what's potassium? Positive. So what happens when you lose positive charge? then it becomes more negative. So this side's called depolarization when sodium's coming in. This is called repolarization when sodium's going out. So this is what you end up with. But here, now we have a problem though, because a neuron doesn't fire one time. A neuron fires many times. Many times. So for a neuron to fire again, it has to go back to this state. So the way it works is it goes from this initial state to this, to this, to this, and then back to this. So how does it go back to this? How does it go from, because if you look at this picture, and look at this picture, they're the opposite. Right? How do you go back to this? There was a little clue, we talked about it. There's a pump. Many pumps that do what? What do you think they do? They pump out sodium while they pump in potassium. That's that sodium-potassium pump. Okay? So the sodium-potassium pump will pump the sodium out. It'll pump the sodium out this way while pumping this potassium in this way. So obviously there's different gates for getting the sodium and potassium through that membrane. Now, let me show you, because the thing is, what you're seeing here is happening in a very particular spot in the neuron. Okay? So, if we go back to what the neuron looks like, remember the neuron, so that, what you saw is actually happening in a particular location. So that is happening over here. So maybe over here you have, maybe the sodium is rushing in, and then the potassium rushes out. But it's not happening here yet. What happens then is as this area undergoes that action potential, it will trigger the next area to do the same. So it travels almost like a domino effect. You knock over one domino and then the next one falls. So you're seeing that in this picture. As one area undergoes the action potential, which is the sodium comes in, right? Then that will trigger the next area to undergo the influx of sodium, which then triggers the next area to undergo the influx of sodium. So one area will trigger the next area. Okay. I don't even remember me putting up this question. It says when there is no action potential, all potassium gates are closed. That's actually false, because not all potassium gates are voltage controlled. But we'll talk more about that later. Okay. Oh, by the way, that whole cycle of from here to here to here to here and back to here can happen 500 times a second. So this is really quick, okay? And by the way, it has to do this because I want you to think back to now in elementary school. When I was in elementary school, no, not elementary, so high school, our libraries had carpet everywhere, okay? And the shelves where the books were kept were all metal. So you know what the guys used to do? And I was one of those guys. We would rub our feet on the carpet, and then we would go up behind someone, and we would like touch their ear. You know why? It would give them an electrical shock. So if you touch something, like if you touch a person or you touch a metal shelf, you rub your feet first and touch a metal shelf, or you get an electrical shock. But if I want to shock that person again, what would I have to do? Because after you rub your feet and you shock someone, and then you touch them again, nothing happens because you have to recharge yourself, right? You have to recharge yourself. So that's what a neuron has to do. A neuron has to recharge itself. And the recharge is quite quick. You can, as I said, do this like 500 times a second. So how long it takes you to say the word nerve is how long that would have, like if you said the word nerve, it takes about a second to say that. That process can happen 500 times. It's quite quick. So, we're actually going to wrap this up very soon. So actually, let's talk a little bit about that refractory theory. So, you'll see in this diagram here that this phase, so sodium going in, potassium going out, you'll notice that this is your x-axis in milliseconds. So how many milliseconds does it take? It's only a few. And then the neuron can recharge itself. So here it's recharging itself, meaning the pumps are pumping up the sodium and pumping in the potassium. So it only takes a few milliseconds to go from trigger back to normal. And so it happens really quick. Really quick. So here, this process, sodium's coming in, and then that triggers the next area. I have a question for you. Why is it that, because you'll notice that the sodium gates here are closed now, right? So initially they were open here on this side, and this triggered this section to open, right? So you can see if you look at the next diagram, you'll notice that now these gates are open. Why do you think this one will open next, but not this one? Why does this, because look, it's closed, and that one's closed. Why does this one stay closed, but that one will open? Why do you think? Because what's happening on this side? What's going on this side? What do you think? Remember I said the one area triggers the next area? So why does this area over here trigger this gate to open, but not that one? Because that one's closed. So you see it's closed. Why doesn't it open? Because what's happening on this side? This area is undergoing it's recharging. Okay? So it's not ready yet to fire. Okay? It's still not ready yet to fire. Okay. Now, let me show you a quick animation then, just to solidify how an action potential works, so you can see it. And then we're almost done, okay? So so here you would have your gates, your sodium potassium gates, and you'll see if we excite the neuron, okay, see what happens. So now we've excited it, so we got to the point where we will trigger it. We will trigger the neuron, and then you can see sodium rushes in, and then that will trigger the next area to allow sodium to rush in, and then this area, you can see, is undergoing repolarization. Potassium goes out. Okay. So what will think? Well, what's going on here? Sodium's rushing in here, so that will trigger the next area to allow sodium to rush in, and then you can see that potassium will rush out on this side. You can see that right here, and then you'll see it again. So it's like one area triggers the next area, which triggers the next area, which triggers the next area. And it just travels all the way down the neuron. Okay? This like domino effect will travel all the way down the neuron until it gets to the end. And then what we'll do tomorrow we'll say, okay, what happens when it gets to the end? We'll talk about the communication between the neurons. Okay? Talk about that part. So the last thing I'm going to mention is why do I have a picture of a pufferfish? What does this have to do with anything? How many of you know pufferfish are poisonous? Yeah? Do you know people actually go to the restaurant to eat, and you can buy them, and why? Why would you eat a poisonous fish? So apparently, not that I'm not a big fish guy, but apparently you can go to the store. I don't know where you can get it here in Canada. I'm sure it is. I don't know if you can get it. Can you get it in Canada?
Yeah, it's professionally made.
Say that louder. You're absolutely right. I was going to mention that. Say that louder. Right. You have to have you got to be professional at doing this, because if you screw up, you can kill someone. The idea is that if you eat pufferfish, if you do it right, you will get this tingling sensation on your tongue, which is supposed to be nice feeling. But if you screw up and you get too much of the toxin, I guess you can go into respiratory distress, and that could be fatal. So what does this have to do with anything? The way the poison works is actually quite interesting. The pufferfish creates a poison called TTX. So 2 T's and an X. Now, what TTX does, and you're going to tell me what you think the effect is. TTX sits in the sodium gate. So it's a poison that will actually sit right in the sodium gate. So it sits in like this. Why is that a problem? So here's my TTX. It doesn't look like this, but it has the right shape that it can perfectly fit. It kind of reminds me of enzymes and substrate. It can perfectly fit into the active site of that gate. What's the problem? What would happen in that case? What would happen in that case? Why would a person then go into respiratory distress? Have trouble breathing? Why? Well, what controls the muscles for breathing? Your nervous system, right? We just looked at how the neurons work. The neurons work by the movement of sodium in and potassium out. What would TTX block? The movement of sodium in, right? That would be a problem. Here's the thing about toxins, right? And see if you can explain this. It doesn't matter if the sodium gates are blocked or kept open. Some toxins will keep the gates blocked. Some toxins will keep the gates open. It doesn't matter. It's the same result. It's bad. They're both bad. You're screwed in both cases. Why? How are they the same? How is blocking a gate the same as keeping it open? So this you guys remember cellular respiration? Remember, to make energy, you have to build a gradient? Well, to run an action potential, you have to build a gradient, right? You have to have a lot of sodium on the outside and a lot of potassium on the inside. If you block the gates, the ions can't move, right? But what's the problem if you keep gates open? What can't you build? Can't build a gradient. In either case, that's bad news. Okay? And we'll talk about how toxins work, because some toxins will block the gates, some will keep them open, some will keep the neurons from communicating with each other. Okay? But we'll talk more about toxins later on. Okay, ladies? So for it today, that is it. That is just an intro to the nervous system. The end.